The Cell Organelle Project by Erica Simmons. The cell membrane's average diameter is 7 μm. Its function is to separate the cell from the outside. It controls what enters and leaves the cell and also recognizes signals from other cells. Its structure is composed of a double layer of fat and a phospholipid bilayer. The cell membrane is present in both plants and animal cells. The cell wall ranges in size from 5 to 100 nanometers. It provides structural support and protection outside the cell membrane and is used as a filtering mechanism and pressure vessel. Its structure is generally tough but somewhat flexible. It is found in only plant cells. The nucleus is typically 6 μm in diameter. It is the control center of the cell and regulates all of the cell's activities. It protects DNA, and its structure is protected by a nuclear membrane. It is present in both plant and animal cells. The nucleolus is typically 1 micrometer in diameter, and it is known as the ribosome production center of the cell. It builds ribosome subunits from rRNA and proteins. It is composed of nucleic acids and proteins found in the nucleus. Its structure does not have a membrane, and it is also found in plant and animal cells. Centrioles are generally 200 nanometers in length, and they serve as an anchor for the cytoskeleton. Centrioles help in cell division by forming spindle fibers. The centriole structure is made up of microtubule proteins, and some float freely in the cytoplasm while others are attached to the endoplasmic reticulum. Centrioles are found only in animal cells. Ribosomes, on average, are 25 nanometers in diameter. They are considered the protein factories of the cells, and they read instructions to build proteins from DNA. The ribosome's structure is composed of rRNA and proteins. Ribosomes are found in both plant and animal cells. The Golgi apparatus is generally 2,500 nanometers in diameter. It finishes, sorts, labels, and ships proteins. It is considered the UPS of the cell. Its structure is composed of membrane sacs, and it is found in both plant and animal cells. The endoplasmic reticulum's function is to work on proteins. It processes them and helps complete the proteins after the ribosomes build them. The endoplasmic reticulum also creates membranes and helps in synthesis and hydrolysis of many compounds. Smooth ER is typically 150 nanometers in diameter, while rough ER is typically 100 nanometers in depth. Its structure is composed of a membrane connected to the nuclear envelope, and it extends throughout the cell. The mitochondria's size varies. It ranges up to several micrometers. Its function is to make ATP energy from cellular respiration, and its structure is composed of a double membrane. It is found in both plant and animal cells. Chloroplasts are generally 5 μm in length. Chloroplast's function is to make energy and sugar from sunlight through photosynthesis. Its structure is composed of 2 membranes, and it is found only in plant cells. Lysosomes vary in size from 200 to 500 nanometers. Lysosomes' function is to digest food, which is used to make energy, clean up and recycle, and digest broken organelles. Its structure is composed of a membrane sac of digestive enzymes. Lysosomes are found only in animal cells. Peroxisomes are about the same size as lysosomes. They vary from 200 to 500 nanometers. Their function is to break down fatty acids to sugar, detoxify, and produce peroxide. Its structure is composed of digestive enzyme sacs. They are found in both plant and animal cells. Vacuoles vary in size, but generally range around 10 μm. Their function is to move material around the cell, and they act as a storage site for food and water. Its structure is composed of a membrane sac, and they are found in both plant and animal cells. The cytoskeleton is generally 10 nanometers in diameter. It aids in structural support of the cell, maintaining its shape and providing anchorage for the organelles. It helps in regulation, organizing structures and activities of the cells. It is found in both plant and animal cells. The cytoplasm size depends on the size of the cell because it flows throughout the inside of the cell. It supports and protects cell organelles. It is a clear, thick, jelly-like material found throughout the cell and inside some of the organelles. It is found in both plant and animal cells. The nuclear membrane size is generally bigger than the nucleus because it surrounds and protects it. It controls movement of materials in and out of the nucleus, and it surrounds the nucleus and is selectively permeable. It is found in both plant and animal cells. Flagellum range in size, but can be as long as 50 micrometers. Flagellum acts as an organelle of locomotion in the cells and has a tail-like projection that protrudes from the cell body and aids in its movement. It is found in both plant and animal cells. Vesicles range in size from 75 to 500 nanometers. They are considered material transports because they transport and digest cellular products and waste. They have a single membrane compartment and are found in only animal cells.